I begin this presentation reminding you that the term speciation is one we use for describing the formation of new species. And there are two mechanisms that have been proposed for how this can happen. And one of those is going to be allopatric speciation. Allopatric means other country or another country. And so if you choose to use the word another, another begins with an A, allopatric begins with an A, and maybe that can help you remember later on which one of the two mechanisms of speciation we're making reference to. So in allopatric speciation, or another country, there's going to be a geographical barrier separating two populations. Like in the example you see here in this slide, there are two species that are real similar when you look at these animals from a physical point of view, from an anatomical point of view. Antelope squirrels of the genus Amnospermophilus. Some live on the southern side of the Grand Canyon. Others are going to be on the northern side. And the Grand Canyon is going to be a barrier that these animals cannot overcome, cannot go from one place to another. And so as the two species separated by the Grand Canyon continue to change over time from a genetic point of view, mutations can accumulate or gene flow may lead to the loss of alleles. These two species are going to remain separate and continue becoming more and more different from one another over time. So that's the concept of allopatric speciation. I think this is going to be the one concept that is the easiest one to relate to. Separate populations have a tendency to evolve over time because there is no gene flow. Remember that the idea of gene flow is one that is necessary for populations to remain as members of the same species. As long as gene flow is happening in members of one population mate with members of another population, that mixing of the two gene pools prevents the two populations from becoming different. But if you have two separate locations and there is no gene flow, then mutations again can happen randomly in different populations and different species over time, and those changes can accumulate, leading to further differentiation between two species. In the example that I chose over here, this is the flightless cormorant of the Galapagos Island. Except for the wings and the legs, these cormorants are very similar to other cormorant species that you can find even in the Puget Sound area, where you have the double-crested cormorants, and the black cormorants as well can be found there. So what about the wings is different on these animals? Well, you can see that these wings, they just don't look right. Some people describe the wings of the flightless cormorant as atrophied or damaged in some way. And so one can look, for example, at the genetic origins of these underdeveloped wings and can find the mutations that are responsible for this change in the wing structure. So needless to say, the flightless cormorant of Galapagos cannot fly. And because these animals cannot fly over to the continental Americas, they cannot be sharing their genes with other cormorant species that can fly. And I mentioned also the legs on these animals. The legs are an important adaptation for them because being flightless, they still have to eat. And the legs are an important adaptation for diving. Flightless cormorants are amazing divers, and they can dive to depths that other cormorants, which can fly, cannot make it there. And so what may have begun as some cormorants migrating to Galapagos, later experiencing a mutation or series of mutations, has allowed these flightless cormorants to become a separate species, being in another country, in a separate place, apart from cormorant species that can fly. The other concept of speciation, which means, again, the formation of new species, is going to be sympathetic speciation. Sym means within or same, and patric means country. So sympathetic speciation is going to be when two species evolve in the same place, in the same geographic location. This is going to be where populations may have overlapping boundaries, but something has to happen to stop gene flow. Remember that stopping gene flow, no gene flow, is essential for two species to become different from, or from one species to become different from another original species. So how can something like this happen in the same place? Well, one of those mechanisms that can lead to sympathetic speciation is going to be errors during cell division, like mitosis. So imagine that, for example, in mitosis, a cell failed to separate the copies of the chromatids that are made. Then instead of a diploid cell making two diploid cells, ends up making a cell that has all of the chromosomes. That cell would have duplicated its chromosomes from 2 sets to 4 sets, and 4 sets of chromosomes is a chromosome arrangement described as tetraploid. Tetra in mathematics means 4, so tetraploid means 4 sets of chromosomes. If this tetraploid cell was like a germ cell, like a cell that is a precursor for gametes, then when it goes through meiosis, it's going to be producing diploid sex cells. And that's wrong, because what we know is that in general, gametes are haploid. They only have half the number of chromosomes of a diploid cell, which is going to be 1 set. But in this case, because we began with that tetraploid germ cell, the gametes are going to be diploid. And when these gametes participate in the process of fertilization, they are going to produce an organism. And if other organisms are made in the same way, now you're going to have a new species that will be tetraploid. And this is going to be an example of autopolyploidy. Autopolyploidy is going to be when within one organism, this error of mitosis and later meiosis allows for the making of gametes that are going to have extra sets of chromosomes, and the subsequent offspring will have also extra sets of chromosomes. If these tetraploid individuals cannot mate with the original organisms that had the normal number of chromosomes, now they become a new species. When do we see this? It's usually going to be with plants. For example, we have a commercial domesticated species of wheat that would have been the result of an autopolyploidy. Auto means within itself, like a population of wheat plants within itself had this type of mitosis and then meiosis error that led to the making of modern species of wheat, which have extra sets of chromosomes. Sometimes the extra sets of chromosomes can be the result of allopolyploidy, and this is going to be when, for example, you have one diploid plant of one species and another diploid plant of a different species end up, for some reason, crossing. Then you are going to have, if fertilization happens, a new zygote that is going to have chromosomes from one parent and chromosomes from another parent that are not going to be compatible. So when later mitosis happens, there's going to be extra sets of chromosomes. And if meiosis is allowed to happen, then there's going to be hybrids being produced with extra sets of chromosomes. If those hybrids eventually gain the ability to reproduce through additional changes in their genotype, then they can end up producing polyploid offspring, which are not going to be able to mate with the original species, and they are going to become a new species. So allopolyploidy here means that two separate species are going to be contributing to the making of hybrids that are going to be polyploid, meaning extra sets of chromosomes, and those can in turn become fertile and reproductive hybrids, making a new species. So this is one example of how in one place you can have, through errors in meiosis and mitosis, the development of new species. By the way, I want to emphasize that autopolyploidy and allopolyploidy are examples we see in plants. In animals, an offspring that develops with extra sets of chromosomes most likely will not survive and will not be fertile at all. So that's, polyploidy is going to be an example of something we see in plants only. Another mechanism that can lead to speciation in the same geographic location is going to be habitat differentiation. And this time I want to share with you also an example of an animal that I studied for a graduate research project. This wasn't my graduate research that led to my thesis, but just another project that I participated in, looking at the development of jaws in cichlids from the Americas. So we have these kinds of fish we call cichlids. Some of them are from Africa, but we also have some that are native to America. They're usually larger in size, and they're going to have two sets of jaws, the oral jaws or mouth jaws, and there's going to be also the pharyngeal jaws, like at the throat level before food moves over to the esophagus. There is another set of jaws. And in cichlids, the pharyngeal jaws sometimes are going to be more developed or less developed. These pharyngeal jaws are going to be important when these fish feed on snails that have a tough shell. And so with the oral jaws, the shell of the snail cannot be crushed, but with the pharyngeal jaws, those shells can be crushed. And here you see what we call the cichlid, the convict cichlid, Cyclasoma manawensi. And then this convict cichlid is going to have like two different forms. This fish you see here on the top has a deeper head. I mean, look at the distance between the upper part of the head and the bottom of the throat compared to this one over here. It's from the same lake, it's the same species, but what I was studying is the development of these pharyngeal jaws, because that's what you see in greater development on these cichlids with a larger, deeper head. What is interesting is that in lakes of the tropical parts of America, near the shore where you have a lot of these snails that are living there, the animals are going to have this larger head because they're feeding on snails. But if you go deeper into the lake where you see mostly sediments with organic matter that fish can feed on, that's when you're going to see this type of form. And what research is revealing is that there are places where there is less and less gene flow between those fish with the larger heads that are feeding on snails and the fish with the smaller heads that are feeding on sediments. If this trend, if this pattern continues right there in the same lake, it's just that the big head ones are living closer to the shore and the smaller head ones are a little bit deeper in the lake. If that continues, that habitat differentiation, the feeding on one type of diet versus another, may be the force that leads to the development of two different species over time. Another example that can lead to evolution of two different species in one place is going to be intersexual selection. Intersexual selection means selection for a mate of the opposite sex. And we've seen this happening with birds, the example of the peacock, and how females choose mates, males, that have the most beautiful tail feathers. So intersexual selection just means choosing a mate of the opposite sex. In the case of animals, this can also lead to sympathetic speciation, because a female may choose to mate with only certain individuals with a certain physical trait or a specific set of behaviors. This is an example that I've also reviewed. I didn't do research with the funnel web spiders of the Sonoran Desert here in the Americas. Agelonopsis epirus is the name of the the funnel web spider. You can just call it the funnel web spider of the Sonora Desert. And when you go to the deserts, often you find creeks that are carving through the desert. And even though those creeks, for the most part of the year, are going to be dry, there is more vegetation. There are going to be certain trees like willows that can grow there and provide a nice shady relief from the heat of the desert. Well, the spiders that live usually in the canyons or in those creeks that carve through the desert are going to be more shy, more timid. They are not as adventurous in terms of catching prey as the ones that live in the drier and hotter part of the desert. Those are more aggressive. And there's a researcher, Susan Reichert from the University of Tennessee. She's been doing a lot of studies with these spiders, and she's discovering that the females from the more vegetated part of the desert where you have a creek, those females which are shy are choosing not to mate with the aggressive males that are moving from the dry, hot part of the desert over to the cooler, less hot life in the creek. And so if this mate preference of the shy females from the creek avoiding to mate with the rough, tough guys coming from the dry, hot part of the desert, that behavioral preference, this mate preference, can lead to the same species in some places to not have any kind of gene flow and eventually become different species. So intersexual selection, we'll see additional examples of how intersexual selection and mate choice can lead to the formation of two different species in one same location. We will be looking in particular next week in lab at the cases of the anoles in the islands of the Caribbean. And so that's it. That's the two different types of speciation, two mechanisms for the making of new species: allopatric speciation and sympathetic speciation.